My name's Lindsay Edwards. I'm the CTO of a company in London called Relation that does a lot of single cell and machine learning for drug discovery. And it's a real treat to be here, wherever Caroline is. Thank you for the invite, and thank you for asking me to put together a committee to judge the posters. I spend way too much of my time thinking about raising money and whether our compute cluster is going to fall over or not. So it's really nice to spend several days thinking about nothing but science. And a couple of quick comments before we get right to the business. The first thing I would say is the level of science over the last couple of days has been absolutely phenomenal. So just to everyone that's here and to all of the students that have presented the last couple of days, you're doing amazing work, so feel very good about yourselves. We tried to choose on the basis of three different things. So the first thing is originality, so how original is the work that's being done. Second thing is scientific quality. And then the third one is a slightly looser one that I would call something like scientific communication. So the way that you were able to speak about your work at the poster, or just the presentation of the poster, the clarity of the language, the clarity of the narrative, all really important. And one thing I would just add to that last thing is it feels like it's not science and we shouldn't care about it, but my God, it's important. Like if you're good at speaking, you'll get asked to speak. And the more you speak, the broader audience there is for your science. So please, people who you see who are really good presenters almost never got that way by accident. They worked on it. It's worth spending a little bit of time on. Okay, so we have 3 prizes. We have £300. Sorry, dollars. You might prefer £300. Sorry, I couldn't help it. $300. So in third place, we have, I'm getting so old, I can't decide whether I need my glasses on or my glasses off. So in third place is poster 21, an AI cyborg system for adaptive intelligent modulation of organoid maturation. And I think we have Jinhee Zhang to collect the prize. Amazing. Come on. Brilliant. Thank you very much indeed. Thank you. Okay, in second place, and with a prize of $400, it is poster 31, Whisper, Inference of Contact-Mediated Cell-Cell Signaling, and this is for Anurendra Kumar. Great stuff. Thank you. And then in first place, we're hoping that the first authors made it back, by unanimous decision, is poster 25, Mapping the Topography of Spatial Gene Expression with Interpretable Deep Learning, and it's Utsav Chitra. Do we have... Is he... Now I sing. No. Okay. What should we do? We can have a little clap later maybe and move forward. Okay. Anyway, thank you to everybody. It's been a brilliant couple of days. Thank you, Caroline.
Thank you so much, Lindsay, for doing this. Okay. Thank you, Lindsay, again. And thank you to all the poster presenters for all of their work that they put into it and to, enriching this conference with their amazing science. So now we're getting to the final panel discussion. So this panel discussion is going to be on foundation models in biology, so kind of the computational counterpart to what we've seen yesterday, and again going all the way from all levels, from DNA to protein to cells, tissues, and organisms. And We have a treat here for you this afternoon. So I think I will reorganize the talks a little bit so that we start with proteins, and then we'll go on to tissues, we'll go on to drug discovery, and we'll go on to organisms and hospitals and biases, etc., all the way at the end. So we'll start with Sergey Ovchinnikov, assistant professor in the Department of Biology at MIT. His lab studies protein structure and evolution in all levels, so environmental, organismal, genomic, structural, and molecular scales. And so welcome, and we'll have your talk first, and then we'll follow up with a panel discussion with everyone at the end.
I wasn't planning to be first. Thought I'd still have a chance to change some slides, but too late now. All right. Right, so yeah, thank you so much for inviting me to be here today. So I'll tell you guys about some of the work we've been doing in trying to understand what protein and genomic language models are doing. Depending how much time we have, we may or may not skip the genomics. But in big pictures, my group wonders a lot, like, what are big models learning? Like we heard today about like the genome language models, protein language models, but what are they actually doing? Like, what's this black box learning? And part of the reason we do this is because my lab, we don't have that many GPUs. That's why I'm wearing this hat. Hopefully somebody would give us some. But because we don't have that many GPUs, we try to say, well, what could we as biologists or a scientist contribute to maybe this giant field where companies are training these giant models? Like, maybe we could look at these models and see, are they learning new biology? Are they learning statistics? Like, how can we interpret them? What can we contribute in that perspective? So we all know proteins, DNA, RNA. This is essentially a little cartoon picture of a sequence, but when you only have a single sequence, there's not much you can say about it. Like, maybe you know some physics, maybe you know some chemistry, but you're kind of stuck there. But where things become really, really powerful is if you can collect a bunch of sequences from many, many different organisms. All of these sequences here all share common ancestry, and there's some positions that are conserved, some positions that are maybe related to each other due to phylogeny. For example, these two positions are due because of connections here. And some positions are covariant with each other, which maybe tell you something about structure. And one thing we always wonder is, how do we capture these constraints? And more importantly, maybe, how do these foundational models that we learned about today, are they capturing any of these constraints at all? And so how do we model these constraints? A very, very simple model is to say, well, let's look at these sequences and just pretend that there's only 3 positions, 3 possible characters. And you could try to really model a very, very simple model where you take the sequence, one hot encode it, because you need to somehow express blue, yellow, green so a computer would understand. Then you could say, well, I'm going to try to model how the first position connects to the second position, how the second position connects to the first position, so you have like a fully connected matrix. So imagine like a very simple linear layer. And then, of course, there's some positions that may be completely conserved. They're not connected to anything, and so you have some parameter that models conservation. And then finally, all the probabilities at the end should sum up to 1. So imagine we have a very, very simple linear layer where you have activations at the very end, and then you try to minimize the inputs and the outputs, say the inputs should match the outputs. If you train such a model on all the data that I showed a little earlier, like train a model on all this data here, you might imagine it will learn a very, very trivial function of just saying, I'm going to learn an identity matrix of anything mapping to outputs. And so it doesn't really learn anything meaningful. Where things become really, really powerful if you say, hey, let me remove self-connections. So I'm going to make sure like the first position is only connected to all the other positions, but not to itself. And then you might imagine the model actually start to pick up on coevolution, like the fact that this position depends on this one, and these edges might have that interpretation. This model that actually does this, one of these models is called Gremlin. And this is the model that I'll be comparing to, where I'm comparing these genome language models and these protein language models too. And so this model essentially is very, very simple. It just has a weights and a bias parameter. That's all it has. But this model is very, very much interpretable. Unlike that black box that I showed at the very beginning, this one you could actually say, hey, these weights, you could actually extract them, and then maybe take some L2 norm of those weights to compress each of these 3x3 matrices into 1. And now you have essentially a contact map. And just to give you a big picture here, essentially these two connections, these 2 residues are connected to each other, and you can extract from the weights the fact that they are actually connected to each other. And so it's very, very easy to interpret. We know exactly what those parameters mean. And just to give you a real example, here is a protein contact map, stretching out this protein from N-terminus to C-terminus, blue to red, blue to red. All of these contacts you see here are coming from the crystal structure. And if I want to show how like the cyan part interacts with the orange part, cyan, orange, I can look up on this matrix here. Now, if I overlay these contacts that were extracted from just this very, very simple model training, I can extract all these dependencies, and the really exciting part is they overlap on the gray dots. So this is around 2011, people got really, really excited because now without any deep learning, this is a very simple, I guess you say simple machine learning, we could extract these contacts from there, and this could mean that we could start to predict protein structures. And so we know how to do it with very, very simple models, but how about more complex model, like a protein language model that has maybe many, many layers of transformers, and as we heard today from Patrick, like maybe some kind of striped hyena layers, like how do we like interpret these things? Like how do we look inside this model? Turns out there's a relatively simple trick you can do because essentially the input and the outputs are the same. Like you still have one hot encoded sequence, you're trying to recreate the sequence on the other side. Maybe you could do something much simpler where you just try to perturb the inputs and see how the outputs change. But before I go into that detail, just to give you a big comparison, so these are traditional models we used to fit. So for every protein family, we fit one model for every family. One of the things that's exciting about these protein language models like ESM, other I guess, related models, is that they are trained one model for all sequences. And they're trained the exact same way. The only difference is for a single protein family, you train one model, or you say, I'm going to train all the models. I'm going to put the sequence in, I'm going to try to recreate the sequence on the other side. And so one thing we've been wondering is, like, are these two models learning the same thing? Like, is there somewhere inside these parameters, these parameters? And so how do we actually check if that's actually true? And so these protein language models, as I said, trained on all sequences. And so the way we would try to do this is say, well, could we try to map the inputs to the outputs? Because essentially this W matrix or this convolution matrix is essentially just a mapping between the inputs and the outputs, like just a linear layer. And the way we could do that is perturb the inputs. And here's a quick illustration here. So I can go to every single position and try to mutate it to some other character and see how the logits change. So I can compute this delta logits, and I can compute a matrix of every possible input to every possible output, and then the exact same way as I did with my really, really simple model, convert that into a contact map. And one thing that we found that quite remarkable was that if you extract these contact maps or these convolution matrices from these very, very simple models, like Gremlin models, and then do the same thing to a protein language models, the pair parameters that you extract are very closely similar and actually very well correlated to each other. So the parameters actually look very much similar between the two kinds of models. And so this sort of gives us this hypothesis that essentially what protein language models are doing are essentially learning like a library of Gremlin models and effectively acting as a very, very nice or cool lookup and compression algorithm. So this could be both a pro and a con. I guess a pro is like, well, now you don't need to make MSAs. Maybe there's some shared parameters between families. Maybe it could do a better job. Maybe a con, if you thought maybe this model solved the protein folding problem, then you're like, well, maybe it didn't. But still, we were kind of wondering, is this actually what it's doing? Is it actually taking the sequence, looking up some model internally, and returning the sequence? And so we tried to design a quick experiment where we said, okay, is it looking up the entire structure, or maybe it's just looking at motifs? Because proteins have little motifs that are shared across all proteins. So maybe it's not actually just storing entire protein families, maybe just storing motifs. And so Ziddy and my group decided to do some experiments for this. She's sitting around here somewhere. I'll try to find her later. But essentially, we said, well, let's take the protein, for example, and find a motif here. So in this case, there's these two helix interacting with the beta strand. And then we try to add more contacts or remove contacts and see how much of the contacts needs to be able to recover these interactions. And the same thing for, let's say, two very distantly related motifs, like this motif and this motif, far away in sequence space, and if you add or remove contacts, what happens? And so what we find is that as you start to remove more and more contacts, interestingly, this motif is still there until you essentially remove so much of the protein that it's no longer able to. So essentially, we're literally just masking out everything around the sequence, and the only thing the model sees is this little motif, and what we see is essentially we can mask almost the whole sequence and still see that motif. Same thing with the pair, like these two helices, we could essentially remove everything except for those two sequences of those two helices, and we still see the contacts. And so what this suggests to us is that the model is not doing like a full fold lookup, but maybe is doing some kind of motif lookup instead. And so we wrote a paper about this. If you guys want to check it out at some point, you could. So in summary for at least this part is that we do have a way of extracting contacts, and it's very, very powerful. You could apply it to any model where maybe you don't necessarily care about what the inputs are, like what exactly is in the middle of the model. And so since then, we've been now applying this model to other models like EVO, EVO2, and asking similar kind of questions. And so maybe, looks like I still have 3 minutes. Okay, so since we have a little 3 minutes, I'll get into genomic language models. So as you guys heard about today from Patrick, is this really cool new model came out where you say, well, instead of training on protein sequences, what if we train on genomic sequences? So essentially the task is very much the same. You have an input sequence, you mask some of your nucleotides. In this case, you could do it in an autoregressive way. And then you recreate the nucleotides on the other side. And there's lots of cool stuff happening in the middle, but for our purposes, like we said, we don't care what's actually happening in the middle. We wanted to see, does it learn the correct input-output mappings? And one thing that was quite exciting, we saw, hey, it looks like it's capturing RNA contacts. Like you could actually see, if you perturb the inputs, the output logics change, and you could see that, it's, for example, in this tRNA structure, it is indeed capturing contacts. But when we applied it to protein structure, we noticed that there were no contacts. So, I mean, it learned some kind of interesting patterns that kind of look like checkerboards, but the contacts, for the most part, appear to be missing. And so we were kind of wondering, why are they missing? For RNA, it looks to be fine, but for protein, like this is an example from a ESM, the protein language model, and this is an example from EVO where we did the same exact analysis. The reason why this is half blank is because it is an autoregressive model. So if you perturb something at the input, only things after get changed, but things later on don't really change. So that's why it's only half. And so some hypotheses we had was maybe the model is just not large enough. And so we did look at EVO2, and it still seems to be missing contact. So we think maybe that's not quite the only reason. Another hypothesis, like, well, maybe there are other constraints that are overpowering. Like maybe there are protein contacts here. It's just that maybe there's other more stronger constraints coming from other genomic constraints that are sort of overpowering the signal. And finally, maybe we're just doing the analysis wrong. So that's always a possibility. One hypothesis that we're kind of exploring now is maybe it has to do more with the fact that what does it mean to encode the nucleotide sequence for proteins? And so you're probably all familiar, but if you like change the last codon, the last letter of a codon, you essentially stay the same amino acid. But even if you change the middle codon, like the letter of this middle codon, you have to either stay hydrophobic or hydrophilic based on transitions versus transversions. So you can maybe already recreate a lot of the sequence just based on your local signal. Like maybe you don't need to learn contacts. And so one analysis by Yunha, who actually is joining MIT later. So if you guys want to talk to Yunha, she'll be here in June. So she's a new assistant professor in MIT biology. So one thing she tried to say, well, instead of using nucleotide encoding, could we just use amino acid encoding? And when you do that, you actually see, so this is just showing control, 2 proteins, contacts between the 2 proteins. And then when you look at GLM2, you actually see really, really strong contacts recovered. And just to contrast that to like EVO and ESM2, there are no contacts recovered. And so we think part of the reason might be because of that reason. Okay, I think I'm out of time, so I'll stop there. Thank you very much.
Thank you very much, Sergey. We'll do discussion afterwards with everyone. Yep, that would be awesome. Perfect. Okay, so I think we'll move to Faisal next. Faisal Mahmood, assistant professor at Harvard Medical School and Dana-Farber. His lab develops multimodal foundation models of going from diagnosis, prognosis, biomarker discovery, and all of that, particularly in the context of cancer, but also in many other contexts as well. So welcome.
Thank you so much, Caroline, for the introduction and for inviting me to speak. I'll be talking really quickly about some of the work that my group has done over the past few years, and more recently what we've done in self-supervised models, foundation models, to try to address some of these questions from a clinical point of view. Here are my disclosures. So what my group does is that we predominantly try to work with pathology images. When glass slides digitized. So pathology, glass slide is really the backbone of biomedicine, whether it's diagnostic, prognostic, therapeutic response related, but it also has lots of applications in preclinical drug screening, trying to identify toxicology and so forth for drug discovery. So a lot of tools we develop are more generic and can be applied to both. But today I'll be talking specifically about some of the work we do in the diagnostic prognostic space. So once these images are digitized, they are hierarchical. They harbor information at many different scales, and they look more like satellite images. And it's a very old diagnostic modality. It's been around for about 125 years, but its computational analysis is relatively recent. So there's been computational analysis for the past 35, 40 years, but really large-scale computational analysis is relatively very recent, and it's just recently begun to be digitized in hospitals. So when we're looking at these large images from a machine learning perspective, what comes to mind is multiple instance learning. So we can split them into smaller patches and then just use multiple instance learning on top of them. And there's a lot of work that was done training these models end to end, which was computationally tedious. And over time, we started to use these feature extractors where we could extract rich feature representations, aggregate it into slide level representations, and then do whatever form of prediction we want to do on top of it. Whether it's diagnostic, which means you could be looking at detection of disease, subtyping, grading, staging, or if you pose it as a ranking problem, it could become prognostic or related to therapeutic response prediction and so forth. And we sort of used this idea in a number of different studies over the years. We tinkered with this method, adding in attention and other bells and whistles that were being developed in the machine learning community, but then also applying it to more challenging problems like predicting origins for cancers of unknown primary, endomyocardial biopsy detection for heart biopsies, and also building large-scale prognostic models by including histology and molecular information. So coming back to this paradigm, so more recently there's been a lot of work using self-supervised learning to extract richer feature representations. A lot of the previous work I showed was all done using ResNets trained on ImageNet and using those features for training these models. So in this case, we try to develop some of those self-supervised models dating back to 2021. And there are a bunch of conference articles we published just showing how self-supervised models can be used in pathology. And then last year we built these foundation models. One of them was really just a DynoV2 using lots of hospital data, and we learned from DynoV2 that diversity of data is way more important than the quantity of data. And we went out looking at Brigham and MGH, what are the most diverse cases, and built those models. And at the same time, we also built a vision language model, so just by using the COCA framework and contrasting with the corresponding text information to see if representation would improve. And these models, we made these models publicly available, and they've really been downloaded quite a lot around the world and have been used in over 400 different studies over the past year. And I think one reason for this is that pathology data is becoming increasingly more available, and it's easy to just incorporate this data if you have rich representations available. But if we think about this, we also have lots of other multimodal data corresponding these patients. Histology really acts as a diagnostic backbone, but there's also molecular data which has been collected over the past 20 years. And we also do immunohistochemistry, special stains, other kinds of ancillary testing corresponding these histology, these patients, and we have that available corresponding to histology. And we have seen this from conventional computer vision and machine learning that contrasting against modalities improves representation for all the modalities. So that's what we tried to explore in a bunch of these studies. This is from ECCV last year, where we're contrasting with IHC stains, molecular markers to see if by contrasting across these stains we can improve representation, and then we apply it to a bunch of downstream tasks, including survival outcome prediction, as well as just creating these IHC slides. And then same concept, but instead of using IHCs, we use transcriptomic data. So this was sort of a toy example just done on the TCGA, trying to see if in a few shot setting the representation would improve. So we're essentially headed in this direction, where by using more and more multimodal data, we're able to improve image representation in itself by using fewer and fewer samples. So this was first sort of an indication where we saw a few shot improvement on some of these more difficult tasks. This was the first indication that we should scale this idea, and we used basically all the molecular data that was available to us at Brigham and MGH, together with paired histology, to see if we can contrast against them. And because we're not fusing here, we're contrasting against them. We could use molecular data that was collected using a variety of different mechanisms, including the NGS assay that we have at the Dana-Farber Brigham Cancer Center, as well as the transcriptomic assay that's frequently used at MGH. And then applying it to a bunch of different downstream tasks, we found that it improved performance, particularly for some of these more difficult tasks like treatment response prediction, where it's difficult to get these samples, so we have very few samples available. And these are just very, very challenging problems. Predicting treatment directly from histology is a challenging problem. But for the first time, we saw a jump in performance. So we've basically been scaling this. We're working together with other institutions to get more corresponding molecular data to scale this idea in general. So once we had these rich feature representations, an obvious next step for us was to see if we could build a co-pilot or a generative model which can synthesize text corresponding this information. So to do that, you need a large instruction set. So the philosophy behind this is that these images are hierarchical, harboring information at many different scales, but the corresponding label that we have just resides in pathology reports, and the label is often corresponding just the slide or all the cases that are all the slides that are in that particular case. But to build a good, useful co-pilot or a chatbot or multimodal large language model, we need instructions corresponding every level of abstraction, whether it's at the region level, at the patch level, slide level, or the case level. So we started by curating those manually, but quickly realized that it was just not scalable, despite asking so many pathologists to contribute to this across multiple institutions. And eventually, we figured out that the best thing we could do was to use teaching and training materials that we had at Brigham and MGH, essentially using what's used to teach residents to teach the multimodal large language model to be able to build this chatbot. But this is a study from June of last year where we're able to build a chatbot where you can go in and ask different questions about the image. In the beginning, this was just based on 500,000 instructions. We did try to maximize for diversity, trying to represent every indication that existed within our Brigham and MGH databases. But since then, it's been really expanded to around 5 million cases, and it also got FDA breakthrough device designation recently. There are better demos for this available publicly. We're also very interested in building agents, particularly for computational biology, but I think this is something that everyone's interested in because generative models are getting better and better, and they can make good scientific plans for what to do. And in computational biology, what we're doing very frequently is bringing new kinds of data to existing tools. We develop a lot of new tools, but a lot of discovery can take place by bringing new kinds of data to existing tools. So the question is that can you start with like a good just a prompt from a user, break it down, have a planning agent that breaks it down into a schedule, and a DAG scheduling unit that schedules which tools to use when and where, and then essentially have a language agent and a coding agent that interfaces with this to eventually create an overall agentic workflow. So this is what we have done just for these tools right here, and this is just a demo of that where the user comes in and says that, well, here I have a cohort of responders versus non-responders, and can you go train a model that would distinguish between the two, and it can go make a plan and then go and execute it. This is surprisingly very easy to build technically because now we have all these models that generate very good plans. More recently, we have updated this to where you can go back and sort of edit what the plan should be, and we have deployed this at MGH and given it to all our pathology colleagues so they don't come to us when they need to train a model, and they can just use this kind of a setup to do so. So once the model is trained, it would you can look at how well the model is doing just by looking at the AUC curve, and then you can go forward by looking at things like the interpretability at the level of the heat map, and maybe that's not good enough, and you can then ask it to describe what it sees within the high tension regions across your entire test set, and it can use our generative model to describe that. So yeah, we're continuing to work in this direction, and then there are a lot of interesting questions when you give them tasks, and we're trying to assess how well these models do in achieving certain tasks, and we're realizing that the models are good, but they don't quite match a graduate student. And the reason is that graduate students are more hungry, and we're trying to assess that how do you build in this this ambition, hunger into the model. And this is like an open question. Other people are working on this too. But I do want to touch upon Apollo. This is a new model that we recently built and would be coming out very soon, where we have used data corresponding 10.1 million patients from Brigham and MGH, and really have tried to use all of the patient record that was available to us. This includes pathology, diagnostic reports, clinical and laboratory tests, and all the EMRs. And we spent about 2.5 years cleaning this data. It was definitely made easier once all these LLMs were available and could be locally served. But the idea is, and this is just a distribution for all the diagnoses that were used, and then we have the overall architecture, where we have built lots of unimodal models corresponding these modalities, and then we integrate them together, and also have a temporal component to this, where there's an essentially a representation which gets updated from based off of more and more information that comes in corresponding these patients, and there's a temporal trajectory to it. And then we apply it to lots of downstream tasks, including for early diagnosis, treatment response prediction, biomarker discovery, clinical trial matching, and we're working with several pharma to try to see how well it can work on clinical trial matching and population health in general. But really, the scale of the data that we have makes this quite interesting, because we have accumulated data not only from Brigham and MGH, but we also have data from other institutions around the country to really integrate these data sets and build a single model that can do patient-level representation. So I want to thank everyone who was, there are a lot of MIT students who worked on this over the years, as well as all the funding that is probably going to be stopped very soon. So thank you so much.
Thanks for ending on such a positive note. Okay, so now we'll take it to drug discovery. Shirley Liu, I'm very excited to have her here. She was professor of biostatistics and computational biology at Dana-Farber and Harvard School of Public Health, where she developed a lot of statistical and machine learning models for many important biological problems, and also actually one of the first single-cell RNA-seq foundation models. And then in 2016, she co-founded GV20 Therapeutics and has been its CEO since 2022. So welcome, Shirley.
Great. Thank you, Caroline, for the kind invitation. Okay, great. Yeah, so as Caroline mentioned, I've been at GV20 for just over 3 years now. If you look at the last decade, I would say cancer immunotherapy, CRISPR, and AI are the most exciting scientific breakthroughs. And at GV20, our mission is to cure cancer by integrating cancer immunology, CRISPR or other kind of high throughput technology and data, and AI or computation. And so we focused on cancer immunotherapy because it is the only treatment to achieve true cure in metastatic tumors. And if you look at the last year, the highest selling cancer drug are the the anti-PD-1 therapeutics. And anti-PD-1 is approved in over 40 indications because they don't just specifically target cancer. They try to boost the patient's own immune system to indirectly cure the cancers. But even for a drug that's like the most, highest sold cancer drug, it only benefits about 20% of the cancer patients. So 80% of the cancer patients still needs better treatment. The conventional approach for a drug research and development process, like an anti-PD-1 or PD-L1 antibody, is like this. It's from gene to function to target or to drug. So first, scientists need to know that there is a PD-1 gene or protein. It interacts with PD-L1 to induce a T-cell dysfunction. Then they can develop an antibody to block this interaction to stimulate the T-cells for cancer killing. And the drug discovery, the target discovery is like 3 and 10 years. Drug discovery is another many years, and clinical development, just overall very long and expensive. So what if I tell you that humans' own immune system actually already produce and optimize anti-tumor antibodies in their tumors? And in fact, over millions of years of evolution enable the human immune system to recognize the tumor targets and optimize antibodies against them. And so they are usually made within the tumors in these tertiary lymphoid structures or B-cell aggregates. And it's been widely reported that the presence of these B-cell aggregates or tertiary lymphoid structures are associated with better patient response and anti-PD-1 response. And so you wonder, if they already have anti-tumor antibodies, why do patients still develop cancer? You can imagine in a lost territory in a war, like a tumor, there are still guerrilla warriors that are still fighting. And if we can have tens of thousands of tumors to look at, even if we don't know why they are still persisters, we know we need to train more soldiers like this to really win the war. And that's the kind of approach we're taking. We have a proprietary AI platform that can identify novel cancer drug candidates directly from these tumor-infiltrating antibodies. We actually don't have to do the tumor sequencing ourselves. There are a lot of published tumor RNA-seq data available out there. And when people sequence the RNA of a tumor, the B cells infiltrating that tumor are also making the antibody RNA. And so we have a computational approach called the TRUST that can assemble antibodies from these tumor RNA-seq. And so at GV20, we have assembled over 100 million of these tumor-infiltrating antibodies from tens of thousands of tumors. They are usually coming from the clonal expanded B cells that are more likely to be recognizing some tumor antigens. Turns out we could use AI to predict the targets of these antibodies, and from there we get to the drug. So intuitively, how do we make predictions? We don't actually train on a known antigen antibody database. Instead, we learn all the features directly from our tumor antibodies. For example, from the tumor RNA-seq expression, you can tell which are HER2-positive tumors and which are HER2-negative tumors. And if you see any antibody features, these are, short peptide features that are enriched in the HER2-positive tumors but depleted in the HER2-negative tumors, that's a potential HER2 binder. And if you have an antibody sequence that have all the right features, then you know it's a potential HER2 binder. We don't just do this for HER2. We have, first of all, a huge tumor collection, and we test it on every gene in the genome and every mutation that's recurrently occurring in the tumor. And so at the end, for every antibody, we have a probabilistic assignment of this antibody to the protein or genes or mutations. And so I hope this kind of convinced you that we can get a potentially interesting antibody. How do we get to the targets? The first thing with this AI approach we tested was that do patients make make a HER2 or EGFR antibodies? These are drugs that sell for about $1 billion a year. And indeed, we can make predictions and experimentally validate that. And then we ask, how about the PD-1, PD-L1? We can also, actually in-house, we have very, very good PD-1, PD-L1 antibody. The PD-L1 we get, we don't even need to optimize. It works from the bat. And then we ask, how about the CD47 or Siglec-15? These are antibodies that have failed in clinical trials. Interestingly, we don't see any patient antibody signals in them. And so we thought, maybe we can use antibody signal to tell us which targets are promising and which ones are not going to make it. And so we use the antibody signal to help us rank the targets. In addition, once we have the targets, we can also use AI to directly get to the antibody drugs. And so that's why we call our approach this STAT, or simultaneous target evaluation and antibody discovery. And so then we said, okay, let's try to predict something new that nobody knows. And we focus on this immunoglobulin superfamily protein because currently all the immune checkpoint genes belong to this family. They are known to mediate cell-cell interaction. And we found that this IGSF8 gene is interesting. Many patients made a little bit of antibody against it. And we also looked at our in-house CRISPR screen data. GV20, when we started, was based on the CRISPR screen technology. My lab was also among the earliest to use CRISPR screens for oncology and immuno-oncology studies. And we found that this IGSF8 gene normally does not influence cancer cell growth unless the cancer cell is under immune pressure. For example, when we co-culture the cancer cell with an NK cell, or when we implant the cancer cell in a full immune mice. And so without understanding the function of this gene, we just said, let's use AI to pull out the antibody that patient made against IGSF8. And fortunately, this target is conserved between human and mouse. So the antibody we got can bind to the mouse IGSF8 as well. So we tried on mouse tumors right away, first in melanoma, but also in lung cancer, colon cancer, breast cancer, and prostate cancer. And we consistently see the same phenomenon like what we observe in melanoma. This drug always have monotherapy efficacy and very often better than anti-PD-1 alone. And when we combine the two drugs together by halving the dose of each, we get even better efficacy. So with this, we kind of felt, okay, we have to understand how this gene works. At that time, when we first saw this gene, there were less than 10 papers ever published on this gene and little known reported function. And so we did the biology function of this gene at the company. This is also why I left Harvard to join the company, because my lab cannot work on it. We published a biology study on the first half last year. We just submitted the second part on the dendritic cell last weekend. So this gene is kind of like a PD-L1, but for the innate immunity. It suppresses the natural killer cells and the dendritic cells, especially when the tumors are losing their MHC class I antigen presentation. So they are usually higher in the immune cold tumors. And they are, because when the tumors don't express MHC class I, they're invisible to the T cells. But they should be recognized and killed by the NK cells unless the cancer cell overexpress IGSF8. And also, it is expressed on DC as well and interact with dendritic cell to suppress the DC antigen presentation and the T cell activation. And so with this antibody, we can directly activate NK cells and dendritic cells and indirectly activate T cells. So it's a very, very exciting mechanism. At the same time as we work on the biology of this target, we brought this drug to the clinic. And we have by now finished the phase 1 monotherapy dose escalation. This drug has excellent developability and really good PK profiles. We have dosed 42 patients, and 38 are evaluable. The drug is very, very safe. And we saw tumor shrinkage in non-small cell lung cancer and cervical cancer. But the very first melanoma patient we tried was a partial responder. And so we asked the sites to give us more backfill of the melanoma patients. And so by now we have dosed 17 melanoma patients with monotherapy. And interestingly, well, all of them have previously been treated with anti-PD-1. 16 has been treated with anti-CTLA-4. And then we treated our drug, just monotherapy. And out of the 9 patients who previously never responded to anti-PD-1, with a primary refractory to anti-PD-1, we saw 3 confirmed partial responders and 3 patients with tumor shrinkage, and another patient who was on wheelchairs before, after being treated with our drug, walked again. And so it's really benefiting the patients. This is from the CT and PET-CT scan. You can see the very first patient or some representative tumors. This big foot lesion shrank significantly. And the second and third patients both have liver metastasis. This one shrank by 58%. And the third patient, the 24 millimeter liver metastasis completely disappeared. This patient, before our drug, in 7 months, used 5 different drugs and progressed very quickly. And initially on our drug, the patient tumor was still growing, and then it started to respond at month 7. And the patient has been on our treatment for over 13 months now and still responding. In comparison to other IO drugs, this drug in the early clinical study is actually better than CTLA-4, much, much better than LAG-3 and TIGIT, because they don't really have any single agent efficacy. In fact, it's similar to an anti-PD-1 from BMS. for the Merck drug, that out of 30 patients, they did have 5 responders, but these patients only had 2.5 line of prior treatment, whereas in our phase 1, together with BMS, it's like 4 prior line of treatment. In immunotherapy, every prior line of treatment reduced the response by about 10%. And so we saw that our drug was actually having similar efficacy as anti-PD-1 in phase 1 clinics. We also saw that the tumor have significantly increased NK and T cell infiltration, even among patients who don't respond to the drug, suggesting that these patients could be resensitized to anti-PD-1. And also the responders seem to have higher IGSF-8 expression than the non-responders, suggesting that we should select the IGSF-8 positive tumors to enrich for responding signals. So how does AI really help us? If you look at the development of anti-PD-1, we don't actually know when the research started, but the very first publication was in 1992. And it took another 8 years for people to figure out the interaction partner was PD-L1. And then another 6 years to develop the drug and 3 years to do the antibody phase 1 study. Whereas from the first day we saw this little known gene was end of like 2019 to the end of phase 1 was only 5 years. And this is because AI told us how patient is fighting the tumor without us understanding why. And in actually a drug development trajectory, if you look at the revenues generated on the Keytruda drug, towards the later years, that's when the income really comes. So if you can shrink the time from patent to drug approval, it can significantly increase the drug patent value. And we are doing this again to build a robust pipeline. In addition to this drug, we have other antibodies, and recently we had a partnership agreement with Mitsubishi Tanabe on some other programs. Again, first-in-class novel targets that we have antibodies for. And so how are we going to use AI? Actually, the AI we use currently is just a deep learning model. We can use foundation models by pre-training on all the proteins and all the antibodies in blood and fine-tune on the antibodies from tumor or from other diseases. But this takes a lot more money. Right now, we need more resources to do these more computationally intensive exercises. But look forward to collaborating with all of you guys, potentially to find a cure for cancer. Thank you very much.
Wonderful. Thank you very much, Shirley. And now it's my pleasure to introduce my faculty colleague, Marzyeh Ghassemi, who is associate professor at MIT in the Department of Electrical Engineering and Computer Science, and also in the Institute for Medical Engineering and Science. Her work is highlighting how many of our current algorithms can really fail patients. So we'll go to the darker side again. But then she also provides new methods for the ethical and reproducible use of machine learning in health. So welcome.
Thank you. There's not a timer, is there? There is.
Oh, it's counting down already? What?
Okay. Hi, everyone. In case you didn't know, there's a large timer that's already counting down just to scare us. And so I'm going to talk a little bit about my research. My lab at MIT is the Healthy Machine Learning Lab, and we really focus on trying to construct methods that will work well in healthcare. In some cases, you don't actually need a model. Maybe you just need to understand that there's some inefficiency. And in some cases, if you build the very best model possible, if you don't deliver the advice correctly, the best model and the best person don't equal the best behavior. So thinking about that at a higher scale. The demonstrative example I always give people is the simple one because I think it illustrates the issue. Let's say that you want to build a system for triaging patients who maybe have a chest x-ray And if they're not sick, you'd really like to send them home early. You don't want them to wait around. And we can build triage models. Triage models are actually reasonably easy to build these days because every person who's ever built an AI model has gone through this nice pipeline where you pick a problem, collect data, define an outcome, develop your algorithm, and then potentially deploy it. Right, and so we can do that too. We did do that. We chose this demonstrative example with our faculty colleagues, and we took the 3 largest publicly available chest X-ray data sets in the United States, 300,000 or over 700,000 images. You can train a dense net to try to predict the finding, no finding, which is the person is healthy, they can go home. And then we get a nice performance, right? Which is what everybody does, right? Everybody makes these really great models. I do this too, that have these really nice performance metrics. The question is, how and when should we be deploying these models? There are many, many models that have been developed for a variety of medical tasks across the range of different data sets in both predictive and generative settings. And when you look at even just one, let's say here we focus on medical question answering, which is a popular benchmark task for machine learning conferences, we see that models are very performant, right? And this is across both closed and open source models, using a variety of different techniques, anything from pre-training, fine-tuning, and just prompt tuning, okay? And it's not just that these models perform well, it's also that we know that there are ways of officially having models out there for use in clinical settings. So for example, the FDA can clear AI algorithms to be used in a clinical setting. So let's look back at my algorithm, right? Maybe this performant algorithm is something, because it gets state-of-the-art performance, we'd like to deploy. But the question is, often we just look at these sort of aggregated metrics, right? And we're not thinking about how that might apply in a wider, more heterogeneous setting. So for example, let's look at the false positive rate of this really, really performant model in different groups. And a false positive rate here would mean that you're underdiagnosing a patient because you're falsely positively saying that they're healthy. And our great model has the largest underdiagnosis rate on female patients, young patients, Black patients, and patients on Medicaid insurance. And if you look at patients who exist in an intersectional identity, they're doing even worse, right? This is not unique to this setting. And really, there's a variety of reasons that these things happen. And there's also a variety of ways that we can try to address them. But it's something to be both aware of and cautious about. So I want to talk briefly about how this happens in medical data, because I think at this point, hopefully we're all aware of how this happens in natural images. Has everybody heard of a shortcut? I'm seeing nods, but I gave a lecture earlier today where people gave me that face. And so I'll just briefly say, a shortcut in natural images is when we know that this is a model, a very nice, well-trained model knows that this is a cow and this is a benign mole, but suddenly this cow is an orca and this mole is cancerous. And it's not because of the actual underlying features we might want it to use. It's because this model has never seen a cow traipsing around in water. It thinks that, large black and white mammals in water are orcas. And also, maybe it's learned a shortcut that, well, I know that when dermatologists mark the area around a mole, they're looking for growth, they think it's cancerous, right? These are things that you didn't explicitly tell the model not to learn, and so it learned it because it's statistically true. It's correlated in the data that it's seen. And this is really funny when we see that frisking cows in water are orcas. It's less funny when we think about features in medical images that maybe might be used to mispredict in certain people. It's even less funny when we think about things that machine learning models can see that we can't see. So if you take a medical note and redact all dimensions of demographics like self-reported race, human doctors can't predict that about a patient. We tested them. But machine learning models can with almost perfect performance and even higher performance when you take a chest X-ray or any other kind of medical imaging. Radiologists cannot look at a chest X-ray. We tested them. They thought they could. But radiologists, they are very confident. Radiologists cannot predict these things, these demographics, but machine learning models can with extremely high accuracy. So what happens when you have a model that can predict something perfectly, a shortcut perfectly, that humans can't see? Well, what can happen is that it can use that shortcut to mispredict more in that group. So here we wanted to explore this in 3 settings, in dermatology, ophthalmology, and radiology. These are all settings where we use medical images to make a diagnosis, and where we have AI models that do these tasks. What we found is that in all cases, models learn to recover information about demographics that in many cases would be very hard or impossible for a human doctor to do. And the more that they recover this demographic secretly in their layers, the worse they are at predicting the outcome for a minority patient group in that data setting. So put another way, the more I know who is female in a chest x-ray, the worse I do at predicting cardiomegaly in female patients. There are ways to fix this, by example, for using robust optimizers, things like group distributionally robust optimization, where you can get this, if you haven't seen a Pareto plot, economists love them, and one of our reviewers was an economist. So here you can see the overall AUC, right? We want that to be high, of course. On the y-axis, we have the fairness gap, and we want that to be low, of course. And you can see when we use vanilla optimization, we get these teal models that are up here for ERM, very undesirable. It's like lying, right? It's saying good overall performance, but the gap is very high. And if we use robust optimizers, we can shove that all the way down. This is great. But this is when we take data from Massachusetts, train a model from Massachusetts, and then we test it in new data from Massachusetts. What happens when we test it in new data from Stanford? It gets much worse. And the problem is it doesn't get much worse uniformly. We actually aren't able to predict whether a model will do much better or much worse in a new setting. And that's a surprising new result. Usually we believe that the best model in one setting will sort of, it'll be worse, but it'll be still the best model of all the models in a new setting. And this is not something that we can get around. It's something that you have to evaluate in new settings. So TLDR1, human data is tricky. Test models on data from populations they'll be used on. The second thing I want to talk about is how we actually optimize models in healthcare settings. Well, there's a hot topic about how we use group attributes. Don't talk to doctors about it, or they'll hit you or yell at you, one or the other, or both. And so the problem is often when you use a demographic attribute like here, I've used sex and age. Even if I use no fancy AI, just a logistic regression model, this model is 1% better on average, but it gets that by being much better in the young female subgroup and worse in these 2 other subgroups, right? And so again, this happens when we have some sort of optimization that is tuning for an overall performance and not respecting maybe interactions between features. And while it's easy to point that out in a logistic regression model, it gets much harder when we deal with these large vision language models, right, that have billions of parameters. And so vision language models are used for things like generation, image classification, and retrieval, but they also have a lot of biases. And the way that people try to debias these models is often this really linear thing where you sort of estimate where is male, estimate where is female, and then you project out and collapse that dimension. But the problem is that assumes linearity, and these are really complex topological objects that are nonlinear, and so if you collapse male and female doctor, you might do bad things to the space for male and female pilot or male and female manager, right? So it has downstream effects that are hard to quantify. So a lot of what we're doing in this space is trying to understand how to work with this complex nonlinear topology. For example, with negation. Despite their flexibility, if you ask models, vision language models, to give you something with negation, they fail. So if you say, I want tables with no chairs, you're going to get chairs. If you ask for a picture of a dog not on grass, you're going to get grass. If you ask in visual question answering whether there is grass, it'll say yes, but there's not. Sidebar, did you know that the most dog-like dog is a golden retriever? If you look at the embedding space for dog, all the other dogs are very far away from it, and golden retriever's right on top. So we have trained vision language models that golden retriever are the only dog out there for us. And maybe it's cute that in vision language models we get these pictures of dogs on grass, but it's not cute when I want evidence of lung opacity and I retrieve this image, but I say evidence of lung opacity with no signs of edema, and I still get this image. Right? It's very important that we process negation correctly with medical vision language models. This is very hard because when we train large vision language models, how do we train them? We train them like this. We always train them on was is present. We don't, for example, say black and white dog jumps over bar. No helicopter. There's never a negation in the caption. And so consequently, vision language models don't know how to contextualize that. And again, this is much harder in medical vision language models because negation is really present, right? Often we just have a list of things that aren't present, lots of negation. And so we made a benchmark to try to understand how poorly state-of-the-art vision language models do in negation, and it turns out it's really poorly. In fact, they tend to embed things right on top of their negations. Not a flower is right on top of flower. And so takeaway number 2, robustness is hard. You have to test models on things that they might fail on, even if humans might not. I see my time is almost up, so I will breeze through the last section, which is about deployment considerations. The goal is not to have AI do everything, hopefully, unless you talk to Dr. Oz, that's what he wants. And so I know, he's like, agents everywhere. I'm like, isn't that the Matrix? Isn't that what the Matrix was? And so really what we want is doctors working with AI together, right? But if that's what we want, that's what we need to measure. And there's lots of good work that has shown first, the losses of bad AI, this is not my paper, but I love it, it's from 2020, is much worse than the gain from good AI. So number 1, make sure that you have a good AI model, right? That's the base, and not all of the models out there are good. Number 2, humans are extremely susceptible to incorrect advice. This was even expert radiologists when we measured. Okay, and so it's very hard for humans to dismiss incorrect advice. We're really gullible. And finally, convincing advice delivery style worked really well on doctors. So when we told doctors to do something bad as an order, they did it. When we told them to do the same bad thing as a factual a feature, they ignored it and retained their original fair decision making, destroying my life work of making better models. So all of these things are really important. These are sort of the human factors that affect whether a model will be useful or not, and they're understudied. So we really need to understand that humans are fallible. You have to test models and humans in these high-risk situations beforehand. And by doing that, we can move forward with ethical AI and health. Thank you.
So thank you to all the speakers for some really, really wonderful and thought-provoking talks. So I would like to ask them all to come up here so that we can have a discussion up here. And hopefully we have a lot of microphones. Okay, so I think this was really, really wonderful going through, from proteins all the way then tissues, drug discovery, and then all the way to hospital, possibly applications and seeing where things can go wrong. So I think we have seen really amazing positive examples of, the use of ML and where we can get to, and then also some scary examples. And so I think I would love to hear from you of we see the complexity of biology and human health. And in some problems, it seems like it's still, we're still very, very far away from making progress on these very complex problems using AI. And others are totally in reach. So how do you go about deciding and which problems do you maybe think that the ones who have very positive examples of, what are the problems that are totally not in reach? And what are the problems that are kind of in reach on your end? Anyone who wants to start?
All right, well, I guess I'll start. Hey, everyone. So I think for us, we're kind of hoping that these giant large language models will at some point start to generalize in terms of start to understand how proteins actually fold and so on. At the moment, they're still in this status where they're essentially storing statistics of what evolution has tried. But if you want to go beyond what evolution has done, how do we? So that's sort of it's still an ongoing challenge, and we're still thinking how to get these models to do it, or even if it's possible at all. Maybe the way we're training them is not the right way, or maybe the architecture is wrong. So I'm not sure yet.
So when I was a PhD student at Stanford, at the time we were taking Daphne Kohler's AI class, and she was discussing how Deep Blue beat the Kasparov for chess playing. And it took another 25 years for general AI models to really benefit everybody. I feel like for AlphaFold 2 to beat other computational biologists in protein structure prediction is the deep blue moment. We are still quite far away from a general AI foundation model for biology, for the whole of biology. And so I think now it's really important to know, especially as a company for us, how much we need to invest in AI at the same time to ensure that the company delivers value and move the company forward. And I think we are still a way to go, but at least, for example, right now we feel like the available data on tumor sequencing is sufficient. But for other diseases, for example, neuroscience or Alzheimer, we are certainly very interested, and there certainly is also an immune component. Something in the brain, maybe the immune system is causing the disease. Or maybe there are still residuals to fight the disease, but we simply don't have enough data there to make drug predictions yet.
So I think that these models are really susceptible to all kinds of failure, just like Marzyeh showed. And we have seen it in real time for some models that we have deployed over the years and have assessed it on actual patients, and we've also published some of those results from a pathology perspective. But I think there are ways you can make them more robust, and I think that some of the more easier problems, for example, in pathology, we're very interested in having a universal triage which can triage out all the negative slides. It happened for pap smears for cytology in 1998, and there's been an FDA-approved tool since, and it decreased the amount of work that was needed. And technically, we could do that in pathology. It's been difficult to get FDA approval for it, and that's because it has very uncertain failure modes. On the other hand, I think that the larger models, foundation models that integrate multiple kinds of clinical data, are really valuable discovery engines, because you can discover a lot of combination markers that you may not have thought of otherwise for all kinds of complex disease, and also to serve as companion diagnostics together with certain drugs.
I think there are really exciting examples of where machine learning, specifically AI generally, is being applied to problems that we're really bad at. So, for example, adverse events in cancer, immunotherapy, those are things we're bad at, right? We don't know how to do it. We're trying to do something new and improve. There's a lot of interesting applications I've seen in women's health where everything is trash, and so if you make any advance, you're doing great. I think the issue is that's not where a majority of the effort is going in a lot of AI for health more broadly, right? I think we're very benchmark focused, right? And so if you look maybe beyond the cancer space to more broadly where AI is already deployed in healthcare, right? A lot of the examples are administrative examples, things that are adjacent to clinical care, like insurers making decisions about who gets different kind of care, deciding who gets booked for appointments, deciding who gets resources, writing notes on your behalf, writing messages on your behalf. And I think those are sort of dangerous use cases because they mediate who is even able to access care even further. So I think there are a lot of really exciting things, but as we all just said, like, we need more data, we need more data. What if we only get data from certain kinds of people, even more so now that they're predicted that they shouldn't be part of a trial, or they shouldn't get to see a pathologist, or they shouldn't, like, those are very scary, I think, examples.
And since now this already came up, we need more data, or, what are really the bottlenecks to making progress? I mean, one could be data. You also all mentioned we need GPUs. For us, like, building the large-scale AI models would require much more funding, even in industry. You also mentioned the other way around as well, although GPUs are there, but maybe you cannot run the lab anymore. So what are, though, the real bottlenecks, you think? I mean, yeah, it could be that. It could be new algorithms. What is it, and which types of problems?
It's capitalism, guys, okay? we all know that you all and the companies, if you're here, I see you, right, that you have the data, that it's been de-identified via HIPAA and you bought it. I know because my friends who work there and are making millions call me, okay? We don't have millions. We can't buy the same data. It would be nice if we could, right? And so I think one thing that would be really helpful is if we did something as a nation, like invest more in All of Us, which is a fantastic platform that I'm a huge fan of. A lot of the work that is done, a lot of the really leading, really cool work that's been done in the past decade is always done first with the UK Biobank, because they invested in that resource. But I think we want to have similar resources across the world. Of course, also GPUs.
So I think that we know that there are three things that you need to do this kind of work. You need the data, compute, and the talent. And there's an abundance of talent in this town, it's needless to say. But for data, I completely agree. We need no large national consortia. I think the UK Biobank is a very good example. They did a really good job at it. I mean, All of Us is great, but it's not as standardized and integrated as we've seen with UK Biobank. There are really large efforts. So I think ARPA is leading some really large initiatives, unless they lose funding too. But they have the index cohort where there's a very large-scale goal of making very large amounts of data just publicly available. And the motivation is that the TCGA was a 10,000 case example, and so much research has come out of just using the TCGA. Everything in my field, in computational pathology, all the initial work, the method development, the initial studies, discovery, was all done on the TCGA, and that was just 10,000 cases. And I think under the umbrella of something like ARPA, they have the ability to just digitize all the glass slides, for example, and the rest of the data, if you can de-identify it, is easy. And then in pathology, we also have the Joint Pathology Center from the military, where they have the largest repository of pathology data going back to the Civil War. And they are digitizing it very, 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 very quickly, and it's a national resource. I think part of their goal is to de-identify that data and make it available for research. So I think that would happen. The compute. So what we have done in the past is that my group had grants that supported compute. Sometimes it was just us, and in other situations it was together with other investigators. And the S10 awards, and they were supporting it, state awards. But I think that if you look at all major institutions, a lot of compute support has come from philanthropy. So we need more of that, and we have that at Harvard. And I think just because these infrastructures, they evolve so quickly, and how much compute you need to develop some of these models. I mean, a lot of the lobbying politics behind building the model that we built using so much patient data was how do we get access to the compute and data and just organize it across many of the Harvard hospitals or a number of years. Yeah.
I would say one bottleneck or barrier to get better or good data is the pressure to publish high-profile papers. For example, tumor RNA sequencing is really inexpensive. You can do it with a couple $100, but people are not doing it. Why? You get another few hundred samples, you can't even publish a CNS paper. And yet, if you do a newest spatial transcriptomics data with 10 samples, you can get a Nature Science. And so I remember Bill Kalin once said that sometimes journals are interested in seeing a mention of hay instead of a house of brick. We need more house of brick type of data to really train these really valuable data to train good AI models. And because these really mention of, hey, the technologies are still evolving. Like you have a little bit of here, a little bit of other samples. They can never really be integrated together to generate really useful insights. And you see batch effect.
Yeah.
All right, guess it's my turn. Yeah, so I actually thought the last talk was quite interesting because I was trying to connect it back to the stuff that we do in terms of fairness. But when we think of fairness in proteins, it's more like there's certain proteins families that actually the models really struggle on, like they don't understand and don't learn anything about. I don't know if fairness is the right word for it. But
they're the worst. We hate them. Those protein families don't deserve to be recognized.
Yeah. And it is one of those things that we often like check. We say, well, do these models, like they start to work really well for large protein families, like where there's a lot of data, but then for small protein families, very undersampled, they don't work so well. And yeah, I think there's a lot of interesting.
Joking aside, it's the same effect, right? It's there's a different dynamic that needs to be captured, right? And if it's an underrepresented sample, it's hard to capture that effect. So it really is the same problem.
Yep, makes sense.
Cool. So I would like to, since we don't have that much time left, to open it up for questions. I'm sure this was very thought-provoking, and there will be a lot of questions. Maybe I'll give this one.
So I have a question on when for all these foundational models for omics, they are coming. And it was good to see the talk about what are the biases. And also in the morning started with Anshul's work where he was talking about TNF biases and stuff. So as a student, it seems with all the foundational models, there are many biases. But as a community, I'm not sure we understand it. It seems, apart from authors, people may not understand where the things. So, I mean, what's your take on, as a student, it seems that there should be some consortium or some symposium dedicated to figure out in what scenario these foundational models, what are the issues with these foundational models rather than what great thing these do. And what's your take on it?
Yeah. I mean, I think that there should be. So let me answer more seriously. I think that it would be really valuable if we had a slightly more invested regulatory landscape, right? I think, for example, if the bill passed by the past administration on AI safety and equity would not have been repealed, or if there were something to replace it, that could be more encompassing and cover foundation models in all settings, health settings and research settings, et cetera. Work on understanding AI bias is actively being defunded. I know MIT faculty members whose NSF careers were canceled because that was their area of focus. Everybody said the foundations will the philanthropy will step up. They have not, I promise you. Everybody said companies will step up. Sorry, they have not. And so this research is actually uniquely hard to do right now. There is very little funding, and if you do that research and you have grants on other things with federal agencies, your grants with federal agencies on those other things might also be canceled. So this is something that I do think we should do a lot of investment into, but it is not being funded right now. So it's hard to do in an academic setting. The play is to rebrand. This is what I was told this morning. It's not fairness, it's safety, okay? As long as we can get the EA crowd to buy in and agree that when AGI comes for us, they'll kill women first. Makes sense, right? Then we can all be AI safety people and get some of that sweet FTX money once they come back.
Thank you.
Anyone else?
So one thing I've noticed is a lot of the discussion about data generation for these models is a little bit agnostic in the sense that actually, as many of you showed, it's actually quite feasible to go and figure out where the models do poorly. So you may not actually, if you just go to the wild and collect data, you may actually be collecting targeting the wrong density of the landscape of data. So do you know of, I don't know as many, but do you know of efforts that are actively looking to fill holes? Model-based data sampling strategies, data generation strategies? Wouldn't those be more effective than, but I guess if all of us is, what is the target objective?
They're oversampling for groups that are underrepresented in other studies. That's sort of their contribution.
I guess in those kind of scenarios, it's maybe easier to tell, but in biological foundation models and things like that, it's very hard to know a priori what it is, but you can iterate through. It's sort of like active learning, but with a big human in the loop. Many years of data collection in the loop.
Yeah.
It just doesn't seem like there are big efforts really pushing. I mean, people have been saying this for decades, but I've just not seen big push in that direction.
Yeah, I think one issue with biological data is that there isn't enough standardization similar to what we get in medical data, right? So a lot of people are complaining about how unstandardized all the medical data is, but biological data is often collected for studies. It's like a hodgepodge of like what it was actually used for. And when it comes to training these foundation models, you want to use every spatial transcriptomic sample that you can find in your entire institution, right? It was done using a number of different techniques. It's collected for different purposes. Was done to answer completely different questions. So the distribution of the data would be sort of all over the place. So in medical data, we have some hope that we know from all these studies from the large companies that diversity of data is way more important than the quantity of data, and it's just really difficult to find diverse data for more biologically oriented problems. So I think what would happen is that there are a lot of these foundation models for all kinds of different purposes. The next step is going to be that they're all trained on different distributions of data, and they all represent the same kind of data. So because their inherent training distributions are different, maybe you can use multiple foundation models to see, or a mixture of foundation models, even using something very simplistic for your downstream tasks. So that's what would happen in the short term before we have more standardized data collection protocols.
Can I just point out also that we really like it when large foundation models find something new in some settings, and then we slap them and say, don't hallucinate in other settings. And so it's also unclear what properties we want for this kind of like forecasting without support in real data space.
Yeah, I guess I'll add, in the protein world, I mean, we have lots of sequencing going on, and folks have been trying to go to weird places like under the ocean and so on, and just sequencing everything. I think I've seen a study where somebody actually swiped the outside of the space station and found some interesting, weird archaea living there. It's probably, I mean, I don't think it came from space, but probably was. But yeah, there's definitely a lot of sequencing efforts happening in specific, and we still see like every 2 years we're roughly doubling in the number of sequences that are quite different from what we see before. So in the protein world, I think might be okay for now until the funding is cut.
Thank you. Based on what you present, it's more not if we can model the human body and health, it's when. And if we can predict, model, and cure every future diseases, what's next?
I'll take this as the last question.
Then we'll have universal childcare right afterwards. That's the next one.
I guess, I mean, I'm guessing you're thinking human health, but how about like all the other animals out there? So I don't know. We'll move on to the rest of life, I suppose.
In addition to physical health, I think mental health will still always be like, how are we gonna make people happier, right? Like, there are also some physiological things. I'm sure we'll have plenty of good things to work out.
Faisal, something more to add?
No, I completely agree. I think it'll take a while to get there, but yeah.
I don't think it's happening anytime soon. Like, we've got a ways, many PhDs in front of us.
So with this and a lot more work to do, thank you so much to this panel for a wonderful session. So this brings us already to the end of this first annual symposium of the Eric and Wendy Schmidt Center. I really loved it. I loved all the exciting talks, a lot of stimulating discussions, and we started from very basic cell biology, tissue biology, all the way to particular pathology, to AI models that were developed for particular biological applications, to foundation models that are very, very broad. So I hope this meeting was exciting to all of you, and that you were also all able to build new connections and had a lot of different discussions, ML will transform all areas of biology and the medical sciences. And, this is so important to bring this community together to actually really make this marriage become better and stronger. So I would like to end by thanking all the people who made this so enjoyable and really made this conference and workshop come together. So that's the amazing team behind the Eric and Wendy Schmidt Center. So I would love if they could all get up and so we have Jennifer, Sue, Samantha, Letitia, Nadia, Elvira, and Orr. I think they're all there. So thank you so much. This was awesome. And also Giro Studio, who helped us with everything getting through so carefully here. So thank you also. And with this, I know we still have a song. Is that right? There is no song? I'm putting Lindsay on the spot. So then you all have to beg him for a song before we go outside. But with that, thank you all for coming.